Good day, everyone. Have you ever wondered how life maintains its incredible diversity across generation? Well, the answer lies in a remarkable process called meiosis. It is a specialized form of cell division which is essential for sexual reproduction. Now, today, let's unravel the intricacies of meiosis, focusing on the two pivotal stages, meiosis 1 and meiosis 2. Meiosis 1 is where the magic of genetic diversity begins. Imagine a diploid cell containing paired homologous chromosomes, one set from each parent. Now, what we're seeing here is prophase being made. This is where chromosomes condense into visible structures. Homologous chromosomes pair up in a process called synapsis. Crossing over occurs and the nuclear envelope starts breaking down. Now, here is the prometaphase. This is where the nuclear envelope disappears. Spindle fibers from opposite poles attach to kinetochores on each homologous chromosomes, and the homologous pairs begin moving towards the cell's equator, guided by the spindle fibers. Now here comes the metaphase. This is when the tetraeds line up randomly at the metaphase plate. The spindle fibers are fully attached, preparing to pull the homologous chromosomes apart in the next phase. Now we're at the anaphase, where it begins with a critical separation, where homologous chromosomes are pulled apart to opposite poles by spindle fibers. This separation ensures each resulting cell will have one chromosome from each homologous pair. Now we're at the telophase. This is where the chromosomes arrive at the poles. The spindle fibers disassemble and chromosomes may decondense slightly, and each cell now has a haploid set of chromosomes. Now here we are with cytokinesis. This is when the two genetically distinct haploid cells are formed, and the cells now enter meiosis 2, which will split into sister chromatids, similar to mitosis. And now, here we are at prophase in meiosis 2. This is where chromosomes recondense into two haploid cells. A new spindle apparatus forms, positioning perpendicular to the meiosis 1 spindle. The nuclear envelope breaks down again, and no crossing over occurs here.
So, for the prometaphase 2, what happens first is that the nuclear envelope disassembles, freeing the chromosomes. Then, the spindle fibers from opposite poles attach to the kinetochores on the sister chromatids of each chromosome. Next up, we have the metaphase 2. First, the chromosomes line up single file at the metaphase plate, similar to mitosis but now in haploid cells. And last, the spindle fibers are fully attached, ready to pull sister chromatids apart. And finally, for the anaphase 2, sister chromatids are finally pulled apart to opposite poles by spindle fibers, and then each chromatid is now an independent chromosome. This ensures that the final gametes will have a complete haploid set of single chromosomes.
Now here we are at telophase 2. This is where the chromosomes arrive at the poles and start to decondense back into loose chromatin. The nuclear envelope reassembles and the spindle fibers disintegrate. And now lastly, we are here at our last stage, cytokinesis. This is where in animal cells, a cleavage foil pinches the cytoplasm into two. In plant cell, a cell plate forms a new cell wall between the nuclei, and the result, 4 genetically unique haploid daughter cells. And there you have it. Meiosis is nothing short of a biological masterpiece. A meticulously choreographed dance of chromosomes ensures the survival and diversity of life. Though It is only two stages, distinct stages, meiosis 1 and meiosis 2. A single cell diploid transform into four genetically unique haploids, each carrying a reshuffled blueprint of DNA.